type of chromosomal mutation is chromosomal number. And of this lesson, you should know how to state aneuploidy effect on autosomal chromosome and sex chromosome. Changes in chromosomal number divides into aneuploidy and euploidy or polyploidy. Aneuploidy is when in a genome, one chromosome has extra or less copy, while in euploidy is when in a genome, the whole set of chromosomes has extra copy. Let's say we have a cell, a diploid cell, contain 6 chromosomes. In aneuploidy, one chromosome has extra copy, hence the chromosome number is 7. Or one chromosome has less copy, hence the chromosome number is 5. While in euploidy or polyploidy, each chromosome will has extra copy, means that additional of one set copy occur in the cell. Additional of 2 set copy is possible in euploidy. Additional of 3 or 4 and so on copy also possible in euploidy. Aneuploidy divides into 2 types, which are monosomy 2n-1 and trisomy 2n+1. While in euploidy or polyploidy, it divides into 2 types, which are Autopolyploidy and Allopolyploidy. So let's discuss the aneuploidy first. Let's say we have a normal diploid cell containing 6 chromosomes or 3 pairs of homologous chromosomes. Monosomy 2n-1 is a condition in a diploid cell where one of the homologous pair loses its homologous partner. For example, this homologous chromosome only has 1 chromosome, so 2n-1 equal to 5, only 5 chromosomes instead of 6. In trisomy 2n+1, it is a condition in a diploid cell where one of the homologous chromosome has extra copy. For example, this homologous chromosome has extra copy, so 2n+1 equal to 7 instead of 6. What causes of aneuploidy? Aneuploidy causes by non-disjunction during meiosis. What is non-disjunction? Non-disjunction is a failure of homologous chromosomes or sister chromatids to separate and move to opposite poles during gametogenesis in meiosis. For example, non-disjunction of homologous chromosomes can occur in meiosis 1. So, if the homologous chromosomes undergoes non-disjunction, homologous chromosomes fail to separate and move to opposite poles. So, these homologous chromosomes will be passed to the same cell instead of separated to another cell. This will produce abnormal gametes n+1 and n-1. Non-disjunction of sister chromatid also can occur in meiosis 2, where sister chromatids fail to separate and move to different poles and pass to the different cell. So, if this sister chromatid undergo non-disjunction, the sister chromatid will be passed to the same cell. This will produce abnormal gametes n+1 and -1 and also will produce normal gametes. So, non-disjunction in gametogenesis produce abnormal gametes n-1 or n+1. Non-disjunction of anaphase 1 of meiosis 1 is explained as below. Non-disjunction in anaphase 2 of meiosis 2 is explained as below. So, non-disjunction can occur in meiosis 1 and meiosis 2. In meiosis 1, non-disjunction of homologous chromosomes occur. In meiosis 2, non-disjunction of sister chromatid occur. Non-disjunction in meiosis 1 and meiosis 2 will produce abnormal gametes, either n+1 or n-1. Non-disjunction also can occur to autosomal chromosome and sex chromosome. What is autosomal chromosome and sex chromosome? This is a normal human karyotype where diploid cell of human has 46 chromosomes or 23 pairs of chromosomes. So chromosome pair number 1 until number 22 is autosomal chromosome, while chromosome pair 23 is sex chromosomes, where sex chromosome determine the gender of individual. XX is for female, XY for male. What happens if abnormal gamete n+1 or n-1 caused by non-disjunction is fertilized by normal gamete? When abnormal gamete n+1 fertilized by normal gamete n, it will produce a zygote with 2n+1 or Trisomy Aneuploidy individual. If abnormal gamete n-1 is fertilized by normal gamete, it will produce a zygote with 2n-1 or also known as Monosomy Aneuploidy individual. What is the effect of aneuploidy to autosomal and sex chromosomes in human? Aneuploidy effect to autosomal chromosomes in human can be seen by using non-disjunction of chromosome 21 as example. Non-disjunction of chromosome 21 will produce two types of abnormal gamete, n-1 with no chromosome 21 and n+1 with extra copy of chromosome 21. If abnormal gamete n-1 with no chromosome 21 is fertilized by normal gamete n, it will produce an individual known as Monosomy 21. This individual has only one copy of chromosome 21 in a diploid cell instead of two chromosomes. So, the number of chromosomes in Monosomy 21 is 45 chromosomes instead of 46 chromosomes. If abnormal gamete n+1 with extra copy of chromosome 21 is fertilized by normal gamete n, it will produce an individual known as Trisomy 21 or Down syndrome. This individual has 3 copies of chromosome 21 instead of 2 copies only. So the number of chromosomes in Trisomy 21 is 47 chromosomes instead of 46 chromosomes. This is a karyotype of chromosomes in Monosomy 21 individual, where at the chromosome 21 there is only one copy instead of 2 copies in normal. This is an example of karyotype for Trisomy 21 or Down syndrome, where chromosome 21 has extra copy of chromosome, hence it has 3 copies of chromosome instead of only 2 as normal. Non-disjunction of sex chromosomes also can occur in male during spermatogenesis. Male has 46 chromosomes with XY sex chromosome. When non-disjunction occurs to the sex chromosomes in male, it will produce 4 types of abnormal sperm. N-1 with no sex chromosome, N+1 with extra X chromosome XX, N+1 with extra Y chromosome YY, and N+1 with extra sex chromosome XY. Both sex chromosomes is present in a gamete. If abnormal sperm N-1 with no sex chromosome is fertilized by normal ovum N, it will produce an individual with Turner syndrome, which is monosomy female with only one X chromosome instead of 2 X chromosomes in female. So Turner syndrome has 45 number of chromosomes with only one X sex chromosome. If abnormal sperm N+1 with extra X chromosome is fertilized by normal ovum N, it will produce a super female individual where this is a trisomy female with extra X chromosome in its sex chromosome. So there are 3 copies of X chromosomes and the number of chromosomes is 47 instead of 46. If abnormal sperm N+1 with extra Y chromosome is fertilized by normal ovum N, it will produce an individual known as super male. This is a trisomy male with extra Y chromosome in its sex chromosome. So the sex chromosome for super male is XYY. There are 2 copies of Y chromosome and the number of chromosomes in super male is 47. If abnormal sperm N+1 with extra sex chromosomes or carry X and Y chromosome is fertilized by normal ovum, it will produce an individual known as Klinefelter syndrome, where this is a trisomy male with extra X chromosome in its sex chromosome. So the sex chromosome is XXY, where the X chromosome has 2 copies and the number of chromosomes in Klinefelter syndrome is 47 chromosomes instead of 46 chromosomes. Aneuploidy effect to sex chromosomes during meiosis in female. If non-disjunction occurs to the sex chromosomes in female, it will produce abnormal gametes. So female has 46 chromosomes with XX sex chromosome. 2 types of abnormal ovum can be produced when non-disjunction occurs to the sex chromosome in female. N-1 with no sex chromosome, and another abnormal ovum is N+1 with extra X chromosome, where the number of chromosomes in an ovum is 24 with XX sex chromosome. Normal ovum is 23 chromosome with only one X chromosome. If N-1 with no sex chromosome ovum is fertilized by normal sperm with X chromosome, it will produce a Turner syndrome individual. If N-1 with no sex chromosomes ovum is fertilized by normal sperm with Y chromosome, it will produce a zygote with Y-O sex chromosome, where this zygote will die during fetus stage. If N+1 with extra X chromosome ovum is fertilized by normal sperm with X chromosome, it will produce an individual known as super female. If N+1 extra X chromosome is fertilized by normal sperm with Y chromosome, it will produce a Klinefelter syndrome individual. This is a karyotype of Turner syndrome individual. There is 45 number of chromosomes, and the sex chromosome only has one copy instead of two copies of X chromosomes, which is normal in female. Since only one copy of sex chromosome, Turner syndrome is a monosomy 2n-1. This is a karyotype for Klinefelter syndrome individual, where the chromosomal number is 47. The sex chromosome has 3 copies with 2 copies of X chromosomes. So, Klinefelter syndrome is a trisomy 2n plus 1. This is a karyotype for super female individual, where the sex chromosome has 3 copies of X chromosome. Hence, super female is a trisomy 2n plus 1. The number of chromosomes in this cell of super female is 47. This is a karyotype for super male, where the sex chromosome has 3 copies. Y chromosome has 2 copies. Hence, super male is a trisomy 2n plus 1. The number of chromosomes in super male is 47. This is some characteristics that are found in Down syndrome individual. This is some characteristics that are found in Turner syndrome. And this is the characteristics that are found in Klinefelter syndrome individual. How about the other type of changes in chromosomal number, which is Euploidy or Polyploidy? Euploidy divides into 2 types, which are Autopolyploidy and Allopolyploidy. Euploidy is a condition in which a cell or organism possesses more than 2 complete chromosome sets. So, this is a normal diploid cell 2n with 2 sets of chromosomes. Additional 1 copy of whole set of chromosome will causes this cell will have 3 sets of chromosomes, or triploid 3n equal to 9 chromosomes. Additional 2 copies of whole set of chromosome to this cell will causing this cell to have 4 sets of chromosomes, or also known as tetraploid 4n equal to 12 chromosomes. 2 types under Euploidy are Autopolyploidy and Allopolyploidy. The difference between these 2 types are it's either the set of chromosomes come from same species or different species. Autopolyploidy is a condition where a cell has set of chromosomes that comes from same species. So this set of chromosomes come from same species. Allopolyploidy is a condition where a cell contains set of chromosomes comes from different species. This cell contains set of chromosomes from different species. In Autopolyploidy, where the set of chromosomes derive from same species, mating will occur between 2 individuals from same species. One parent undergoes meiotic error where non-disjunction occurs to the whole set of chromosomes in meiosis. So, instead of producing a normal gamete n equal to 3 chromosomes, it produces unreduced gamete with 2n with 6 chromosomes. When unreduced gamete with 6 chromosomes fertilized by normal gamete with 3 chromosomes, it will produce an individual with triploid or 3 sets of chromosomes, 3n containing 9 chromosomes. From 2 sets of chromosomes, now the new organism produced has 3 sets of chromosomes. As a conclusion, fertilization between unreduced gamete and normal gamete may form Autopolyploidy, a condition where a cell or organism has more than 2 sets of chromosomes. Fertilization between unreduced gametes also may form Autopolyploidy, mating between 2 parents from same species, where both parent undergoes meiotic error during meiosis, where non-disjunction occurs to the whole set of chromosomes in meiosis. Hence, each parent produces unreduced gamete with 2 sets of chromosomes each. When these 2 unreduced gametes undergoes fertilization, it will produce an Autopolyploidy with 4 sets of chromosomes, also known as tetraploid, where the chromosome number is increased into 12. Non-disjunction of whole set of chromosomes also can occur during mitosis. Species A diploid cell has 6 chromosomes. During S phase in DNA synthesis, each chromosome undergoes duplication forming sister chromatids. During anaphase of mitosis, sister chromatids will separate and move to opposite poles and pass down to another cell. But since non-disjunction occurs to the whole set of chromosomes in anaphase during mitosis, sister chromatids will fail to separate and move to opposite poles. Hence, all the chromosomes will remain in the same cell. This new cell has 4 sets of chromosomes, 4n or tetraploid. Number of chromosomes increase, which is 12, from 6 into 12. When a normal cell division occurs by mitosis, new cells with same number of chromosomes will be produced. Second type of mutation in Euploidy is Allopolyploidy. Allopolyploidy is when set of chromosomes derived from different species. Species A diploid cell has 6 chromosomes, mate with species B diploid cell with 4 chromosomes. Species A undergoes meiosis producing haploid gamete with 3 chromosomes, and species B undergoes meiosis producing haploid gamete with 2 chromosomes. Fertilization between these 2 gametes will produce a hybrid with 2 sets of chromosomes from different species. This set of chromosomes came from species A, while this set of chromosomes came from species B. This is a sterile hybrid, which is the first generation. Hybrid consists set of chromosomes from different species. By doubling the chromosomal number of the sterile hybrid will be changed into fertile hybrid, which is the second generation. So what happened to the set of chromosomes after chromosomal number is doubled? Now we can see here each chromosome has its homologous partner. Hence, this cell has 2 sets of chromosomes, 2n or 4n, equal to 10 chromosomes number. Why 2n? Because each chromosome is in pairs. Why 4n? Because there is 1, 2, 3, and 4. 4 sets of chromosomes. So you can use either 2n or 4n in fertile hybrid second generation. Why the first generation of hybrid is sterile while second generation is fertile? So sterile hybrid means this hybrid cannot produce gamete because they cannot undergo meiosis. Sterile, also known as infertile. Fertile hybrid means this hybrid able to produce gamete because they can undergo meiosis. Sterile hybrid has no homologous chromosome. Hence, they cannot undergo meiosis. Hence, no gametes can be produced by sterile hybrid. While after doubling the chromosomal number, the fertile hybrid now has homologous chromosomes. Meiosis can occur and gametes can be produced. So, the hybrid in second generation is fertile. As a conclusion, in the chromosomal number mutation, Aneuploidy divides into monosomy 2n-1 and trisomy 2n+1, where one chromosome has less one copy or extra copy, while in Euploidy or Polyploidy, it divides into Autopolyploidy and Allopolyploidy, where the whole set chromosome numbers is changed, either comes from same species or different species.